Hey guys, welcome to Bioinformatics Vaccine Design. Let's start with the introductory part of vaccine designing. What is meant by vaccinology? So, it's a branch of medicine concerned with the development of vaccines. The word vaccinology means science of vaccine development. Okay, so who is the founder of vaccinology? It's Sir Edward Jenner, and what he did was in 1796 he inoculated an 8-year-old boy with vaccinia virus or cowpox, and he demonstrated immunity to smallpox. So this vaccinia virus is the live viral component of smallpox vaccine. So this first vaccine developed, that is smallpox vaccine, in 1798 eradicated smallpox globally by 1979. Now let's see the types of vaccines and the first developed vaccine in each vaccine subtypes. Okay, so the first one is live attenuated vaccine. So live vaccines, what it does is it use a weakened or attenuated form of the germ that cause a disease. Now, because these vaccines are so similar to the natural infection that they help prevent, they create a strong and long-lasting immune response. So just 1 or 2 doses of most live vaccines can give you a lifetime of protection against a germ and the disease it causes. One such example, or the first live attenuated vaccine that was developed, is smallpox vaccine by Edward Jenner. Next is inactivated vaccines. They use the killed version of the germ that causes the disease. Inactivated vaccines usually do not provide immunity or protection that is as strong as live vaccines. So you may need several doses over time, or you can say you may need booster doses in order to get ongoing immunity against disease. The first developed inactivated vaccine is cholera vaccine by Louis Pasteur. The next type of vaccine is messenger RNA vaccine or mRNA vaccine. What they do is they make proteins in order to trigger an immune response. mRNA vaccines have several benefits compared to other types of vaccines, which includes shorter manufacturing times, and because they do not contain a live virus, there is no risk of causing disease in that person getting vaccinated. Example is COVID-19 vaccine. And the next other types of vaccines are, you have subunit vaccines, recombinant vaccines, polysaccharide vaccines, and conjugate vaccines. All these vaccines use specific pieces of the germ, like its protein, sugar, or capsid. Capsid means it's a casing around the germ. So because these vaccines uses only specific pieces of the germ, they give a very strong immune response that is targeted to key parts of the germ. They can also be used on almost everyone who needs them, including people with weakened immune systems and long-term health problems. Now, one limitation of these vaccines is you may need booster shots to get ongoing protection against disease. Now, the first developed subunit and recombinant vaccine is the hepatitis B vaccine. And the first developed polysaccharide vaccine was the pneumococcal vaccine, and Haemophilus influenza type B conjugate vaccine was the first conjugate vaccine that was developed. The next type is toxoid vaccines. As the name suggests, it uses a toxin or harmful product made by the germ that causes a disease. They create immunity to the parts of the germ that cause a disease instead of the germ itself, okay? So which means the immune response is targeted to the toxin instead of the whole germ. So again, like the other types of vaccine, you may need booster shots to get ongoing protection against disease. The first developed toxoid vaccine was tetanus vaccine. And the last type is viral vector vaccine, which uses a modified version of a different virus as a vector to deliver the protection. Example is the Ebola vaccine. So these are all developed by conventional vaccinology. Okay, here what we are dealing is the reverse vaccinology. So what's the difference between both these? In conventional vaccinology, on the left hand side, just focus on that figure. Microorganism is cultivated, then antigen is selected, followed by identification, testing of immunogenicity of this antigen, and component purification. Next, the genes are cloned and it is tested in animals, and then there occurs development of vaccine. Whereas in reverse vaccinology, it's mostly or a mix of dry lab and wet lab. Conventional vaccinology is pure wet lab method. Whereas reverse vaccinology starts with genome sequence analysis, followed by prediction of novel antigens, then recombinant protein expression studies are done, then the immunogenicity testing is done in animal model, and the vaccine is developed. Let's just have an overview about what conventional vaccinology is and what reverse vaccinology. Here in conventional vaccinology, most abundant antigens during this disease can be identified. But here in reverse vaccinology, all the antigens can be identified during the disease, even if it is immunogenic or even if it is not immunogenic. That's the biggest advantage. Then here in conventional, microorganisms has to be cultivated, whereas in reverse, there is no need for it, right? In both the conventional and reverse vaccinology, animal models are essential. Then in reverse vaccinology, correct folding and recombinant expression is important. Also, the high throughput expression or high throughput analysis, even that is important. But the advantage of doing conventional vaccinology is polysaccharides may be used as an antigen, lipopolysaccharide based vaccines can be possible, then glycolipids and other SD1 restricted antigens can be used. But in reverse vaccinology, non-proteic antigens cannot be used. Only protein-based antigens can be used. That is, the polysaccharides, lipopolysaccharides, glycolipids, and other CD1 restricted antigens cannot be used. But the advantages are fast access is there to virtually every single antigen. Then you don't need to cultivate microbes. Then non-abundant antigens can be identified here. Then antigens which are immunogenic during infection or not immunogenic during infection can be identified. Then antigens which are transiently expressed during infection, even that can be identified. Again, antigens which are not expressed in vitro methods, that can be identified. The non-structural proteins can also be used. Whereas in conventional vaccinology, only structural proteins can be considered. And antigens which are not expressed in vitro, that cannot be identified. Okay. Then there is a need for a longer period of time for antigen identification. Then antigen variability also occurs. Okay. So that's the major difference between both these. Let's focus on reverse vaccinology. So reverse vaccinology is a part of immunoinformatics. Okay. Immunoinformatics means it refers to the application of high throughput mathematical and bioinformatics approach to immunology. So by definition, reverse vaccinology is the technique of applying the computational approaches for the development of potential vaccine. Now, the image that is shown is Sir Rhino Rapauli, who is actually the first vaccine developed through this technique, okay, is against serogroup B meningococcus by Rhino Rapauli. So he is known as the father of reverse vaccinology. Now, the other vaccines developed using reverse vaccinology is vaccines against H. pylori, Zika virus, dengue virus, leptospirosis, SARS-CoV-2 viruses, and so on, okay. Now, since I've mentioned that reverse vaccinology is a part of immunoinformatics, let's see the other branches of immunoinformatics other than the reverse vaccinology. So you have B cell epitope predictions, you have T cell epitope prediction, then the other branch is software and database development. Then next you have the chemoimmunoinformatics, which is nothing but the design of drugs against immunological proteins and molecules. Then you have allergenicity prediction and immunogenic or non-immunogenic protein engineering. Then you have antibody, biosensor and adjuvant design and engineering. Then the other branch is vaccine and immunogenic construct designing. Then you have immuno-omics, autoimmunity and transplant, system immunology, which includes cell immunological networks, pathways, and response modeling. Then you have the immunological high throughput data analysis, which includes NGS and microarray analysis. So these are the other branches of immunoinformatics. But here, let's just focus on reverse vaccinology. In the next session, we'll go to the practicals. Okay, that's about it. Thank you.